Hello, I'm Nina, a final year PhD student at the Royal Veterinary College in London, and the title of my poster is the Interpretable Multi-Label Prediction of Mycobacterium tuberculosis drug-resistant phenotypes from whole genome sequence variants. So, to provide some background to the poster, tuberculosis is caused by members of the Mycobacterium tuberculosis complex, and in humans the primary infection site is in the lungs. The Mycobacterium tuberculosis complex is considered an unusual group of bacteria, and this is because it has a clonal population structure with very little ongoing horizontal gene transfer and strong phylogeographical associations, and this is known to confound statistical and machine learning analysis. As with many bacterial infections, drug resistance in tuberculosis is a growing problem. Drug-resistant tuberculosis is typically diagnosed using drug susceptibility tests. However, these can take a lot of resources and a long time. Therefore, more recent efforts have been moving to use whole genome sequencing to profile drug-resistant strains and characterize the mutations that underlie drug resistance, and this has utilized statistical and machine learning analysis. Machine learning has wide applications across the biomedical sciences and has demonstrated high-level performance for genotype to phenotype predictions. A key example of this is the prediction of drug-resistant phenotypes from Mycobacterium tuberculosis genomic variants. However, there are several critical limitations remaining, and these limitations include: firstly, confounding due to population structure. The Mycobacterium tuberculosis complex has lineages with associated mutations that are known to confound machine learning prediction. Single label learning. Most machine learning models predict resistance to a single drug at one time. Multi-label models can predict resistance to more than one drug at a time and take into account label dependencies. And finally, interpretability. It's important to leverage machine learning prediction to draw useful biological insights and clinical adoption. The aim of this study was to develop a novel approach for interpretable multi-label prediction of tuberculosis drug-resistant phenotypes from genomic variants. To achieve this aim, we developed the following machine learning pipeline. We began with 18,396 Mycobacterium tuberculosis samples. In terms of features, we used variants and candidate genes in line with genes in the TB Profiler database, which has undergone expert curation for drug resistance mutations. Our outcome was drug-resistant phenotypes across 10 drugs. We split our data into training data, so 80% was used to train the model, and from there we built a phylogenetic tree and derived a weight, and this was to train our feature-weighted random forest model. The feature-weighted random forest model is a previously developed model that assigns a probability to each feature that will be used as a split variable, and it uses this probability from the phylogenetic tree to help it control population structure confounding. We turn this into a multi-label model using a classifier chain approach. We then used 20% of the data, our testing data, to measure the performance and interpret the model. As we've mentioned, interpretability is a key element of machine learning models, and therefore we wanted to focus on this aspect. We developed knowledge graphs from our multi-label feature-weighted random forest models, from which we derived interaction coefficients to build the edges between nodes of features. We then integrated domain knowledge from TB Profiler, variant type and effect from SNP-F, and also a PPI network or protein-protein interaction network from String. This allowed us to overlay lots of information together in order to interpret our multi-label feature-weighted random forest model to understand the interactions between features and those that might be involved in drug resistance. We wanted to compare the performance of our model to several other models to see how the multi-label feature-weighted random forest model compared to more traditional approaches. You can see here that across all of the drugs, we achieve moderate to high performance in terms of the AUC-ROC. We see that the AUC-ROC is particularly high for rifampicin, isoniazid, ethambutol, and pyrazinamide, and these are the first-line drugs. In terms of sensitivity, the multi-label feature-weighted random forest model exceeded that of traditional approaches. However, for second-line drugs, it was a little bit more limited. The more traditional approaches had higher sensitivity for some of the second-line drugs. However, they were largely dependent on non-causative mutations, which include lineage-specific mutations indicating confounding from population structure, as well as co-occurring mutations, which cause drug resistance to a different drug. As mentioned, a key limitation associated with machine learning prediction that we wanted to overcome was its interpretability. And to overcome this, we built knowledge graphs which utilise information from the multi-label feature-weighted random forest model, as well as domain knowledge. And we overlaid this information to build the knowledge graph and be able to dynamically filter nodes to indicate how likely they are to play in drug resistance. So you can see here we filter the nodes based on how likely they are to play a role in rifampicin resistance. And we can highlight also protein-protein interactions to highlight genes that may also underpin drug resistance as well. In conclusion, machine learning prediction of drug-resistant phenotypes in Mycobacterium tuberculosis has been limited by confounding from population structure, single label learning, and lack of interpretability. We developed an approach which utilises the multi-label feature-weighted random forest model in combination with knowledge graphs to overcome these limitations. We showed that the model is capable of moderate to high performance and that knowledge graphs are useful for exploring feature interactions alongside prior knowledge to improve machine learning interpretability. And this has key implications for clinical settings.